while breeding the new Santoran varieties for cultivated. However, Santoran is more complex. That reason we try to find more information about the genetic to improve. And my study focused to study Santoran and try about the genetics by try to find some QTL by Genome-wide association study. And now I will move to the material and method. I use 1,332 accessions, include indica rice, japonica rice, oat rice, and aromatic rice. Then I conducted it in the greenhouse and tried to evaluate after 2 weeks of sowing. So for the protocol at the sowing time, I also put the different Santoran, 0 and 60 millimole of sodium chloride, and renew the solution after 7 days and 10 days. And after 2 weeks, I evaluate the Santoran by SES score. SES score means standard evaluation score, and we evaluate by score 1, 3, 5, 7, and 9. Among them, 1 is the most Santoran and 9 is the most sensitive. And after getting the phenotyping data, I also will make some genotyping data by download some genotyping data. I remove some amino frequency less than 5% and remove the accessions with the missing rate larger than 55%. And then I got almost 600,000 SNPs. And because it's too large, so I just random selected 40% for genotypic data analysis. The other, I select 1% to calculate genetic similarities, and I analyze the PCA. Then I combine all the genotypic data, genotypic similarity, PCA, S covariant, and phenotyping data, and use the model factor specificity transformation linear mixed model, or we call fast LMM. And then we got some Manhattan plot and some 5, and then we select when we group based on LDDK of each group, and then I got some QTLs of Santoran, and here we refer to SES. I move to the result. We can see in the left side, in the left corner, we have the 3 fingers. Finger 1 is the PCA plot of genotypic data of whole population, 1,332 accessions, and divide in 5 different groups. We can see it's obvious to see the different cluster with the purple color in the left corner is the japonica rice, and the right corner is with blue color is indica rice, and above in the top is the osmotic, or we can call boro. So we see the different groups, and aromatic and admixed is spread out of the plot. So to analyze GWAS here, we need to consider a population structure, and here we use like PCA like as a covariate data. The second finger is the LDDK of different groups. With the red color is the aromatic, and follow with japonica, whole population, ost, and indica rice. So with indica rice, we have the smallest LDDK, so it means that when we consider the size of the QTL is the smallest. And the third finger is about the frequency distribution of SES score in moderate salinity, and here moderate we set with 60 millimole of sodium chloride. Next column is about the Manhattan and QQ plot of Genome-wide associate study for SES score with letter A, B, C, D, E, F. Sorry, I don't know. It didn't appear here. But A is whole population, B is indica subgroup, C is japonica subgroup, D is OS subgroup, and E is aromatic subgroup. And the right side is the QQ plot. So the QQ plot, if we see the QQ plot, we can see the model is well fitted to the data, so we can use this model for the result. And we see the Manhattan plot, and I set the threshold about the, I set the threshold is 4, loss score is 4, so finally we find, and based on the LDDK, finally I can find 12 QTLs. Among them, we have 3 QTL with the narrowed, black narrow is the contain the genes controlling the salt tolerant. And the right side is the table about the QTLs detected, and the more information about 12 QTLs include chromosome column, peak position column, and the next is the whole population indica japonica osmotic, because we didn't see any QTL in aromatic population, so I didn't put here. And the right column is about the information of 3 genes